Our next speaker is Jongtae Kwon, a postdoctoral associate in Gloria Choi's lab. His talk is titled Neuromodulatory Regulation of Social Behavior in Amygdala, and he will be introduced via video by his mentor, Gloria Choi.
Hello, my name is Gloria Choi. I'm an investigator in the Picower Institute. It is my great pleasure to introduce Dr. Jongtae Kwon. Dr. Kwon graduated from Korean University in Seoul with a Bachelor of Science degree in psychology. He then went on to receive a PhD in neuroscience from KAIST before joining my lab as a postdoctoral fellow. I am proud to say that he will now be going back to Korea in about 2 weeks to start his own independent career as a principal investigator. Jongtae has taken a unique role in my lab, which now mostly studies neuroimmunology, in that he continued the tradition of studying olfactory circuits and their ability to modulate social behaviors in mice. He will tell us today about a novel circuit he identified in the amygdala to have an ability to suppress social behavior when conditions are unfavorable, and how this circuit can be reconfigured according to internal states by neuromodulator TRH. So without any further delay, I want to welcome Jongtae.
Thank you. Thank you, Gloria. I didn't know she prepared this amazing introduction of me. Far from there. So again, my name is Jongtae, and I have studied neuromodulatory regulation of social behavior in Gloria Choi lab. So in many different species, including like insect, bird, fish, mammal, of course, human being, lives in a social group because it gives incompatible benefits to survive and maintain the species. But in return, the social individuals also take some burden to deal with the other social conspecifics, right? If you look around your daily life, most of our life challenges are actually coming from the other people relationship. Yeah. So depending on interacting partner or social circumstance, we social individuals have to know how to deal with these situations. So let's think about more innate behavior, such as mating. In laboratory condition, if you actually introduce a female mice to a sexually naive male, you will find that it will take only 2 to 3 minutes for the sexually naive male mice is already on the top of the female. So although this is literally first time to see female, they know what to do, and then it's really strong instinct. But even for this strong behavior, what if the female is in a sick condition? If they has infectious disease, virus or bacteria, would it be still beneficial to mate with the female? Maybe not. So actually, the infectious disease is one of the most critical social risks, as we have experienced recently and now. So even after medical health and vaccines, millions of people have died. And in some cases, infectious disease actually can terminate a social group at a time. So naturally, animals should develop some of ability to detect it and also defend themselves. And if that's the case, that function should be installed in the neural circuit. So our research question was, how does the brain detect the sickness and then regulate our social behavior? So to address this question, we utilized the lipopolysaccharide, LPS, which is a bacterial protein component. So when injected in the mice, it triggers immune reaction and then make the animal sick. So as you can see here, the male subject, 90% of male subject actually shows mating behavior toward healthy, normal female, but only half the male try to mount toward the sick female. And even for those half, their actual mounting time or number of mounts was significantly reduced, which means, yes, male mice also can recognize the sickness and then they change their behavior. How is that possible? We know that olfactory system is important for social behavior. This is particularly true for rodents. And then as you can see here, olfactory system consists of two separate organs. Here's the yellow part is the main olfactory organ that processes most of the volatile chemicals. Separate from that, this vomeronasal organ under the nasal cavity, this is specialized for non-volatile chemical, mainly pheromone. So this organ itself is already quite designated for social stimuli. And then the further project send the information to the accessory olfactory bulb. So we surgically removed this vomeronasal organ from a male subject and then found that these male actually mate with sick female, LPS injected sick female. So we now know that pheromonal information is very critical. And then this pheromonal input through the accessory olfactory bulb further projects to a few brain regions, including beta nucleus, medial amygdala, and posterior medial cortical amygdala. So by conducting immunostaining of the C4s as an indirect marker of the neural activity, we found that actually it's the posterior medial cortical amygdala most strongly activated by sick female. So this was interesting because cortical amygdala, actually there was no study, previous study. There was only, if you search it, there was only one single functional study reporting that this is really not important for mating. And now here we observe that, okay, this is not important for mating, but it could be important for suppressing the mating. So we further investigated its dynamic activity during the social interaction using calcium imaging. And the male subject is interacting with sick female or healthy female while we are monitoring cortical amygdala activity, calcium activity. As you can see in the right side of the graph, this black curve shows the response to the healthy female from the very stable baseline. Once they contact and then sniff the female, it increases the response, evokes the response. But although they keep sniffing and they're following the female, the response quickly habituated and then silent. But in contrast, this orangey curve is the response to sick female. And then this response is initially a lot bigger. And then when they parted from female and approach female again, the response will come back and repeatedly evoked again and again. So we found a very high negative correlation between cortical amygdala activity and actual mounting time. So simply saying, while the interaction, if cortical amygdala activity goes up, then mating goes down. So we further confirmed this by activating and inactivating the cortical amygdala. So for the activation study, we used optogenetics. So by illuminating the light stimulation, we could activate the cortical amygdala when the male mice interact with healthy female, and it totally robustly suppressed their mating behavior, even towards healthy female, only when the light was on. In opposite way, when we chemogenetically turn off this cortical amygdala and then introduce the sick female, the male mouse was still mating with them. So here, up to here, we found that, we found a new brain locus that's important for regulating mating behavior. So now our next question was then how this cortical amygdala exerts its functional role in the neural network. So we traced its axonal projection and found that it actually projects to ventral hippocampus, medial amygdala, beta nucleus, lateral septum. All of these nucleus has been mentioned and then studied in the social behavior. And among these nucleus, we found that the medial amygdala was receiving most intensive input from medial amygdala. So the medial amygdala is interesting. It actually has two different clusters of the neurons. On the dorsal side is packed with VGAP positive, GABAergic inhibitory neurons. On the ventral side, BGL2 positive, glutamatergic excitatory neuron. So previous researchers have found that activation of this GABAergic inhibitory neuron immediately induce social behavior. When these neurons are activated, they suddenly mount or attack the other mouse. Opposite way, if you stimulate this glutamatergic excitatory neuron, Bglut2 neuron, then it stops the social behavior. And then they just show the asocial behavior, such as self-grooming. So in our functional study, while we are stimulating cortical amygdala and then recording imaging the response from this glutamatergic or GABAergic population, we found that cortical amygdala actually functionally activate this glutamatergic neuron. That makes sense, right? So we can think that this posterior medial cortical amygdala respond activated by sick female and further activate the medial amygdala Bglut2 neuron to suppress their mating behavior. So to prove this circuit mechanism, now we combined optogenetics and chemogenetics together. So instead of the stimulation of cortical amygdala cell body, now we here specifically activated the terminal projection onto medial amygdala, or we directly activated medial amygdala glutamatergic neuron and found that in both of conditions actually nicely recapitulated the cortical amygdala activation result by suppressing the mating. In contrast, inhibition of this medial amygdala glutamatergic neuron directly, it also reversed their mating behavior towards sick female. Combining these all together, while we are activating its projection onto MEA that resulted suppressed mating, reduced mating, if we additionally inhibit the medial amygdala Bglut2 neuron, the postsynaptic neuron, then it also reversed their mating behavior, to demonstrating that yes, this is cortical amygdala to medial amygdala projection indeed mediate this mating suppression function. So, but we know that, and that we found that this cortical amygdala nucleus as a new brain locus. But as we know, a nucleus contains many different types of cells, and so we wanted to define what is the genetic characteristic of these neurons. So for that, we had to specifically label these cortical amygdala neurons that project to Bglut2 positive neuron. So for this, we combined pseudo-rabies virus system that for the retrograde monosynaptic circuit tracing with so-called trap method for mRNA purification. For this work, so the Wickersham lab and then Heyman lab, we collaborated with these two labs. It was really amazing experience. So when we encountered this difficult question, our great neighbors come up with immediate and perfect solution, and we carried out it and solved the problem. So it was one of my best experience in Picower Institute while I'm studying. So by doing this, we found that there were multiple genes. Among those, TRH, thyrotropin releasing hormone, was one of the most highly expressed gene in cortical amygdala. So we are familiar with TRH. It's released from hypothalamus, and then through the pituitary gland, it finally activates the thyroid gland, regulating our energy metabolism. But this well-known neuromodulator is now unexpectedly found in this cortical amygdala region in a very high expression and also very confined manner. So again, this is neuromodulator, meaning that it will be released, then the postsynaptic neuron also might have the receptor expression. Indeed, we found the TRH receptor is very highly expressed in the medial amygdala, and it was quite highly expressed with Bglut2 gene, consistent with our previous observation. So what would be the cellular function of this TRH receptor signaling? So to test this idea, we performed the slice calcium imaging. So in the baseline, these are the Bglut2 neurons expressing calcium indicator. And when we applied TRH, 20 to 50 seconds, we could see they shine brightly, meaning that calcium activity goes up, and this is actually excitatory cellular function. So it means that TRH signaling in the medial amygdala might mediate the mating suppression function. So to test that, this time we directly injected TRH into the medial amygdala, and then after this injection, mouse really didn't mate or didn't show like interested with female. So fully recapitulating the cortical amygdala activity, and also found that in opposite way, if we remove, conditionally knock out the receptor, these mice actually doesn't care about the sickness of the female and then mate with them, mount. So as a summary, we found that, okay, male mice also can also care about female sickness. And then this information through the pheromone input delivered to cortical amygdala. And then TRH receptor signaling in the medial amygdala excitatory neuron for the suppressed mating behavior. Now, as a follow-up study, we are further studying the function of this TRH system, a new neuromodulation system. And then it was actually not limited for the sickness detection or mating behavior. We found that it's actually engaged, this system is engaged in more general social behavior, such as aggression or parenting. And then what we found is it seems to encode potentially stressful social stimuli. Actually, sick female is one of the example. And then the other example would be like when they meet the other male mouse, cortical amygdala is also activated. If the interacting male is more dominant and aggressive, the activity goes up. So in other aspects, this TRH system is also affected by their social experience. So we gave like social stress by placing this animal to aggressive male, so they attacked repeatedly for 3 days. And after this, if you test their mating behavior, they are not mating well. And then found that in these animals, TRH receptor is very highly upregulated. So we are still in the very beginning of this study, but in our neuroscience history, we know that uncovering a function of neuromodulator have dramatically improved our understanding of the neural function. So we hope that we can also contribute for the social function. Yeah, so for this study, our lab member, especially the Dr. Changhyun Ryu and Irene, all the other members, and then again, Heyman Lab, and then Wickersham Lab, I appreciate it. And then most of all, Gloria, throughout all my fellow postdoc period, she supported my science and also my life. Thank you so much.
Thank you so much, Jongtae. I think we'll try to do the same thing because I'm not seeing any online questions. Can you see if there are hands raised?
Yeah, sure.
And then and just repeat the question before you answer it. That'd be great. Thank you.
Yes, please.
I'm wondering, does this cortical amygdala region receive any other type of sensory input, or is this specific to olfactory social cues?
So in our study, we didn't find any like specific projection from the other sensory modality. But we cannot exclude the indirect pathway like ventral hippocampus or other nucleus. But yeah, again, the direct projection was mostly from those pheromonal input. Yes, thank you. Yes.
Very nice talk. I was curious if you think that similar amygdala circuits play a role in human social behavior.
Yes. Oh, so okay, that was actually one of my big questions during my study because.
Can you repeat the question?
Oh, okay, sorry. So the question is, oh, he's not here. The question is then this system, cortical amygdala system, is also preserved in human, so would have same function. So basically, human does not have this abomination organ, so cannot be same. But we found that in an old study like 50 years ago, people studied like expression of TRH or TRH receptor in the human brain, but human cortical amygdala and then also highly expressed the TRH, and also receptor is expressed in a similar area. So it might not be exactly the same function, but I think TRH function is still preserved in the human. Thank you. Yes?
I got two questions. Great talk, first of all. Are the same neurons activated by the LPS-induced female and also the aggressive male, I guess? And the second question is, just to be fair, are the same circuitry involved in like females' rejection of the sick males?
Yeah. Okay, the first question is, so I showed that the cortical amygdala responds to female and male, whether these would be same population or not. Okay, I have no answer for that. So for that, we need to measure the single individual neurons, but we didn't do that yet. So that's one of our plan. So possibly or not, but okay, again, that's not, I don't know yet. Second question was, sorry, can you?
Are the same circuitry that you discover from the male brain, is it also conserved in the female brain, such that it might be used to reject the males that's LPS injected or other sickness?
Okay, the second question is, this cortical amygdala system is also, it would be similar between male and female, basically. That's a great question. We don't know. We only tested with male mice. And then if we think about actually behavior aspect, phenotype, it's also different. So again, generally, this cortical amygdala TRH system is for giving some alert about potentially stressful. Possibly for the female, male itself could be stressful sometimes. So that is also one of the research topics that we need to address.
I see one question.
Yeah, seems like time is over. Let me take just one more question.
Yeah, there's one question here that's come in online that I can ask. What is the molecular signal detected by the male mouse? Is it LPS acting directly or a chemical produced by the LPS-treated female? And if LPS is introduced into the environment but not into the female mouse, is mating inhibited?
So if I correctly understood, the question is actually what chemicals is detected by male from LPS injected female. So actually that was one of our questions. We tested mass spec by collecting urine and fecal sample from the female and then tried to find that specific molecule, but failed it. So until now, we have no answer for that. So yeah, but we rather focused on how this neural system support this behavior. I think I took too much of time. Thank you very much.
Thank you very much, Jongtae. And if other people in the room have questions, you can talk to Jongtae during the break.